In this video, we will explain how the structure of the cell membrane aids its function and how substances are transported in and out of the cell. Cell membranes, also known as plasma membranes, are primarily made of a bilayer of phospholipids. Phospholipids are amphithetic molecules with polar heads and nonpolar tails. When forming the cell membrane, the nonpolar tails face each other, while the polar heads interact with either the cytoplasm of the cell or the outside environment of the cell. This is caused by the hydrophobic tails not liking interaction with polar substances. The orientation of the phospholipid bilayer plays a major role in the function of the cell membrane. The cell membrane acts as a barrier, separating the inside environment of the cell from the outside environment of the cell. At the same time, the cell membrane needs to be able to transport molecules into and out of the cell. The cell membrane does this through semi-permeable property, where it only allows certain molecules to be able to travel freely into and out of the cell. The membrane only allows small and nonpolar molecules to be able to cross the membrane freely. Big and polar molecules are unable to cross without the help of proteins, as the nonpolar tails block them from crossing. Now that we have explained how the structure of the membrane affects its functions, we will talk about how molecules cross the membrane. There are three categories of transport: passive, active, and bulk transport. Passive transport is when substances cross the membrane in either direction without the use of energy. This is because the substance is going down the concentration gradient, going from an area with high concentration to an area with a low concentration of that substance. There are two main kinds of passive transport: diffusion and vacillated diffusion. Simple diffusion is for small and nonpolar molecules that can easily cross the membrane without any help from proteins. Vacillated diffusion is for large and polar molecules that are only able to cross the membrane with the help of proteins. Some common proteins that are used in vacillated diffusion are channel proteins and carrier proteins. Channel proteins are only used in passive vacillated diffusion and are faster than carrier proteins. Although they look like they can bring any substance across, channel proteins are actually very exclusive, only allowing a specific type of molecule to cross. Carrier proteins are used in both vacillated diffusion and active transport. The protein changes shape once the target molecule binds to the binding site. There is one special exception, of course, which is water. There is a special term for the diffusion of water, which is known as osmosis. Although water is polar, it is also very small and is able to diffuse across the membrane by itself, which is simple diffusion. At the same time, large amounts of water are able to be quickly transported across the membrane through aquaporin proteins, which is vacillated diffusion. On the other hand, active transport is when substances cross the membrane using energy. This is because the substances are being moved against the concentration gradient, going from an area of low concentration to one with a high concentration. All molecules in active transport require the facilitation of other proteins. We will talk about one type of protein that is actually commonly used in active transport: protein pumps. Protein pumps are basically a subcategory of carrier proteins. A little different, pumps can be triggered by substances other than the one involved in the transport. One example, used a lot in AP biology, is the sodium-potassium pump. The sodium-potassium pump is used to maintain a cell membrane's potential, creating a relatively negative environment inside the cell compared to the outside environment. The sodium-potassium pump does this by removing 3 ions of sodium from the cell and bringing in 2 ions of potassium. Although more sodium leaving the cell than potassium comes in contributes to making the environment relatively negative, the main reason is because the high concentration of potassium inside the cell causes the potassium to leave the cell through a channel and therefore making the cell have less ions. The last type of transport we will talk about is bulk transport. Bulk transport is when larger substances are transported across the membrane using vesicles. There are two main kinds of bulk transport: endocytosis and exocytosis. Endocytosis is when substances are transported into the cell using vesicles. In endocytosis, there are also two categories: phagocytosis and pinocytosis. Phagocytosis is specifically for larger substances such as other cells. Pinocytosis is specifically for fluids and small macromolecules. Now, we will talk about exocytosis. Exocytosis is when substances are removed from the cell using vesicles. To remember exocytosis from endocytosis, think of X in exocytosis as exit. And that is all for this video. Thank you.